Directionality is important in the structure of nucleic acids because nucleic acids are added in a 5' to 3' direction. The 5' end of a nucleic acid is the 5th carbon spot where the phosphate group has been added. So 1, 2, 3, 4, 5. The 3' end of a nucleic acid is where a free OH molecule is found. So for example in this one, 1, 2, 3, this is the 3' end. Nucleotides can only be added in the 3' direction, meaning they can only be added to the 3rd carbon on the sugar molecule. So, for example, should we have added another nucleotide to this sequence, a phosphate group would be added right here. Nucleotides are the building blocks of nucleic acids. Each nucleotide is composed of a phosphate group, nitrogenous base, and a 5-carbon sugar. The 5-carbon sugar found in nucleic acids is different for DNA and RNA. DNA uses deoxyribose sugar, while RNA uses ribose. The ribose sugar contains 2 free oxygen atoms, one at the 2nd carbon and one at the 3rd. Meanwhile, deoxyribose only has 1 free at the 3rd carbon. This is one difference between DNA and RNA. Another is the nitrogenous bases that are used. DNA uses A, T, C, and G, while RNA uses all of these same bases except it replaces thymine with uracil. C, U, and T are pyrimidines, so cytosine, thymine, and uracil, while adenine and guanine, or A and G, are considered purines. A always pairs with T or U in RNA, while C always pairs with G. These bases pair together through what is called hydrogen bonding. The breaking of hydrogen bonds between the nucleic acids causes the double-stranded nucleic acids to unzip. So when it unzips, it would split directly down the center. When A and T or U pair up together, there are 2 hydrogen bonds, as seen here. So between these 2 right here, there would be 2 hydrogen bonds. C and G pairs contain 3 hydrogen bonds. So should we look at this at a more molecular level, it would have 3 hydrogen bonds between it. Some other bonds that exist in RNA and DNA are phosphodiester and glycosidic bonds. Phosphodiester bonds occur between the 3' and 5' carbon of 2 sugar molecules. The phosphodiester bond, a covalent bond, creates the backbone of the DNA/RNA, giving it its strength. Glycosidic bonds occur between the sugar and the nucleic base. So for that would be between this and the 1 carbon on the sugar molecule. Glycosidic bonds are important because they help provide stability to the overall nucleic acid.